Welcome to Easy Microbiology. Good morning everyone. Today we will be discussing Enterococcus gallinarum, gram-positive bacterium belonging to the Enterococcus genus, which is commonly found in the gastrointestinal tract of birds, including chickens and turkeys. We will explore its habitat, morphological features, biochemical characteristics, host range, virulence factors, mechanisms of antibiotic resistance, and more. Let's take a closer look and begin.
Enterococcus gallinarum is a gram-positive bacteria with coccus morphology, belonging to the Enterococcus genus. It is commonly found in the gastrointestinal tract of avian species such as chickens and turkeys. This species is notable for its resistance to environmental conditions and many antibiotics, and has been identified as the cause of a range of diseases, including colobacillosis, in poultry and other birds. Enterococcus gallinarum is a type of bacteria commonly found in the intestinal tracts of birds, particularly chickens, turkeys, and other poultry. This bacterium can also be found in soil and water environments, indicating it is widely distributed in nature. It is important for poultry industry professionals to be aware of this bacterium, in order to protect the health of their birds and improve overall biosecurity measures.
Enterococcus gallinarum is a gram-positive bacterium that has a spherical or ovoid shape. It is typically found in pairs or short chains and does not produce spores.
Enterococcus gallinarum is a bacterial species with versatile growth capabilities, able to thrive under aerobic and anaerobic conditions, across a wide range of temperatures and pH levels. Microscopic observation of colonies reveals them to be small, round, and grayish-white. By paying attention to these characteristics, one can learn to optimize the environment to maximize bacterial growth or inhibit it. Gallinarum is a facultative anaerobe, meaning it can survive both with and without oxygen present. It can ferment glucose, lactose, and sucrose. It is positive for catalase, pyrrolidonyl arylamidase, and leucine arylamidase. However, it is negative for oxidase and indole production. Enterococcus gallinarum is an opportunistic pathogen which is commonly present in the intestines of poultry, including chickens, turkeys, and other birds. Although its activity is mainly concentrated on birds, it may also be transmitted to humans and animals. Thus, precautionary measures must be taken when in contact with poultry. To avoid any potential contamination, it is necessary to handle and cook poultry products adequately. This slide is about virulence factors of Enterococcus gallinarum. The bacterium is known to produce several virulence factors which allow it to easily colonize and infect host tissues. These include aggregation substance, cytolysine, gelatinase, and hemolysin. This plays an important role in its ability to cause infection and disease.
Enterococcus gallinarum is a bacterium that has become resistant to multiple antibiotics. This includes aminoglycosides, beta-lactams, and vancomycin. Resistance can occur through different mechanisms such as the acquisition of plasmids, transposons, or chromosomal mutations.
Epidemiology looks into how diseases are distributed among a population. Enterococcus gallinarum is a common microorganism living in the digestive tract of poultry. It can result in illnesses such as bacteremia, endocarditis, and urinary tract infections in both humans and animals, especially those with weaker defensive systems. Therefore, it is essential to take suitable precautions to evade infection. Enterococcus gallinarum bacteria can cause infection through a process known as pathogenesis. This involves the bacteria producing various virulence factors that make the tissue susceptible to infection. Different strains of the bacteria may produce different virulence factors, allowing them to infect a wide range of host tissues and cause various infections.
When it comes to Enterococcus gallinarum infections, the host immune response is activated and involves different types of immune cells. Neutrophils, macrophages, and lymphocytes are among the cells that play a role in fighting off the infection. Additionally, the production of antibodies and cytokines is stimulated to enhance the immune response against Enterococcus gallinarum.
The bacterium Enterococcus gallinarum is a cause of infections in humans and other animals, and it is diagnosed by culturing the bacteria from clinical samples such as blood, urine, or wound swabs. Once the bacteria have been cultured, diagnosis can be confirmed using morphological, cultural, and biochemical characteristics. Additionally, molecular techniques, such as PCR, can be used to identify the organism. Good hygiene practices in poultry farms and hospitals are essential for preventing Enterococcus gallinarum infections. Additionally, antibiotic stewardship programs will help limit the emergence of antibiotic-resistant strains. These programs manage the use of antibiotics to reduce resistance and minimize the risk of infection. Antibiotic treatment of Enterococcus gallinarum infections may require the use of penicillin, vancomycin, or linezolid. However, it is important to carefully select the most effective antibiotics to reduce the possibility of antibiotic resistance. Our study of Enterococcus gallinarum has come to an end. We have established that this gram-positive bacterium is commonly found in the gastrointestinal tracts of birds, such as chickens and turkeys. It is capable of causing a variety of infections, and antibiotic resistance is a serious concern. It is imperative that good hygiene practices and antibiotic stewardship programs be implemented if we are to prevent and control this pathogen. Thank you for your attention and time.
If you enjoyed the content and found it helpful, please consider liking the video and subscribing to our channel for more informative content like this. Your support is greatly appreciated.